Dear viewers, greetings. In this video, we are going to see about Coagulase test. Aim. The aim of the Coagulase test is to differentiate Coagulase positive Staphylococcus aureus from other Coagulase negative Staphylococcus species like Staphylococcus epidermis and Staphylococcus saprophyticus. Principle. Coagulase is an enzyme that works in conjugation with normal plasma components to convert fibrinogen to fibrin and causes clotting of plasma. This enzyme is a good indicator of pathogenic potential of Staphylococcus aureus, as 97% of the strains of Staphylococcus aureus have proven to be coagulase positive. The Coagulase test is useful for differentiating Staphylococcus aureus from other gram-positive catalase negative cocci. Staphylococcus aureus produce two types of coagulase. First one is bound coagulase and the second one is free coagulase. Slide coagulase test is done to detect bound coagulase, whereas the tube coagulase test is done to detect free coagulases. Both slide and tube coagulase test utilize rabbit plasma treated with anticoagulant to interpret the normal clotting mechanism. Bound coagulase. Bound coagulase is also known as clumping factor. Bound coagulase is attached to bacterial cell wall and reacts directly with fibrinogen. And this is shown by formation of visible mass. It does not require coagulase reacting factor or CRF. Next, free coagulase. Free coagulase is an extracellular enzyme which is released from the cell. Free coagulase converts fibrinogen to fibrin by activity of coagulase reacting factor in plasma. Coagulase reactive factor is also called as CRF. This is detected by appearance of fibrin clot in the tube coagulase test. It is usually recommended to do tube coagulase test on all slide coagulase negative staphylococci. Materials required for the coagulase test are test bacteria, saline, rabbit plasma, glass slide, test tubes, inoculation loop and incubator. Procedure for slide coagulase test. First, place two separate drops of saline on a slide and then using a sterile inoculation loop, emulsify 1 or 2 colonies of organisms in one drop to make a thick suspension of bacteria. After that, add a loopful of plasma to both the suspension and saline drop and mix gently, and after that look for immediate coarse clumping of the mixture within 10 to 15 seconds. Procedure for tube coagulase test. Dilute the plasma as 1:10 dilution with saline and take 2 test tubes and add 0.5 ml of diluted plasma to each. And then inoculate a tube with bacterial colonies to make a cloudy suspension. Alternatively, add about 5 drops of thick 18 to 24 hours broth cultures. And after inoculation, incubate both tubes at 35 degrees Celsius for 1 to 4 hours in a water bath. Afterwards, examine both tubes for presence or absence of clots. Finally, observation and results of slide coagulase test. In slide coagulase test, a positive reaction was observed by formation of macroscopic clumping in 10 seconds or less in coagulated plasma drop and no clumpings in saline or water drop after addition of a loopful of bacterial culture. In negative reaction, no clumping in either drops. And next, observation results of tube coagulase test. In tube coagulase test, a positive reaction was observed by clot of plasma drop at any size after addition of loopful of bacterial culture, but in negative reaction, no clot was observed. Dear viewers, thank you for your support. Thank you.